Hello, it's good afternoon now, everyone, and welcome to the last day of the UK DNA Working Group and EOF webinar week. And we're joined with our final group of speakers that I'll introduce throughout the course of the afternoon. And so to start off with, I'll introduce myself. My name's Cy Kriya. I work in Bangor University and one of the coordinators of the Molecular Ecology and Evolution group here. And I'm also supported by Kitty Pillay today. So Kitty, would you like to introduce yourself?
Hi everyone, my name is Kitty and I'm a PhD student with Cy at Bangor University. I'm looking at mangrove fish in Florida, looking at the connectivity using metabarcoding and stable isotope analyses.
Okay, so we all know why we are here. We've seen various webinars throughout the week, and so we're going to finish with our last academic and research focused day. I'd like to remind everyone that there is a hashtag associated with the presentations, and I see people using that. It's really useful to get messages out on the Twitter sphere. And also the webinar software is telling me that we're being recorded. So these meetings will be recorded and we'll endeavour to make them available on our YouTube channel after the webinars have finished. And so just briefly, today we're going to be joined by Florian, Christy and Naira and Christine. They're all going to talk about their ongoing research programs and views of the field. Then we're going to have a small wrap-up talk by myself. And then at the end, we'll give everyone a chance to join in with the panelists to ask any broader questions that may or may not have been asked throughout the week. But for now, what I would like to do is pass my screen over to Florian, and just while he's getting ready, I'll just introduce him. And so Florian's given me a lovely bio. I'm sure many of you know that Florian did his PhD between 2004 and 2008 in the Alfred Wegener Institute and the Helmholtz Center for Polar and Marine Research, working on invertebrate biodiversity in the Southern Ocean. And since then, Florian's done a bunch of postdocs in Ruhr University in Bochum. And then since 2015 and now in 2021, he's moved through the ranks of W2 and W3 professor of aquatic ecosystem research. But I thought a really nice suggestion from Florian was, you can ignore all of that and just know that he's a guy that wants to join everyone together when it comes to accelerating the uptake of eDNA technologies to enhance biomonitoring for the future. So thank you very much, Florian, and I will melt away into the background. And if I can ask the other panelists to do the same, that would be great, and over to you.
Okay. Yes, thanks, Cy, for the introduction. Glad to be here. I don't only have good news, I have a couple of not so good news. They're also reflected in my title, which reads Back to the Future, National Roadmaps hopefully pave the way to harmonized international biomonitoring 2.0. And I'm absolutely against national solo actions. And on the other hand, I see that at the moment we have to go that way somewhat. But I think there's also a positive twist to it. Let's have a look. First, I deleted all my introduction slides, and I think it's much better to use the tweets that actually came out with the hashtag of this DNA working group meeting to show where we actually stand. And I think it's quite clear. Nobody has to tell us that it's a boring research field. It's amazing what you can do with all the different DNA and eDNA techniques. We can improve environmental monitoring by combining artificial intelligence with DNA-based methods. We can go beyond eDNA and study eRNA to look at physiological stress by just tracing the molecules the organisms release. Yes, we all know eDNA is not the holy grail. If we use eDNA methods, we know there's stuff like primer bias, etc., etc. But I would say we have also good solutions to many of the problems. And I fully agree with this statement. we don't have to demonstrate that there's good value of the methods for improving environmental monitoring. We are far beyond that step. Everybody knows it. It's not perfect, but it's really valuable. And what we also see, yesterday, we had the talks by Alice, by Seb, by Kat on their amazingly good companies that actually support decision makers and users by actually generating eDNA data if they don't have labs themselves. So my point will actually link directly to this statement. And let me just, I have so many windows of the webinar control pane here, so now I can read it. So this tweet by Andika was really good. She says, or she summarizes one of the presentation days by saying, the trend of eDNA is clear, more and more hip work, but let's now focus on its application rather than demonstrate how fancy it is. And the legal frameworks are vital. And this is exactly what I will show in my talk. We have these legal directive ecosystems. So may they be international instruments for monitoring our environment, or may it be European instruments, and of course we have regional instruments and national ones. The basis for all these instruments typically are observational data. So Water Framework Directive, Marine Strategy Framework Directive. The aim is to assess ecological or environmental state. And this is based on taxonomic data. So collect samples and by comparing to reference conditions, assess whether it's in a good or a poor or whatever state. And then there are thresholds that shall not be crossed. Typically, it shall not be below good. And the vision that the network DNAquanet has, I introduced it actually at the UKDNA working group back in 2016. I was in Edinburgh. That was a great meeting. I have very good memories on it. So I introduced it. It now just closed, and I will talk about this a little, because our vision was to actually link the legal frameworks that exist with the novel technology. And we did not succeed everywhere. But briefly to show the network. So this is kind of the core group below, the working group leaders, etc., and the management committee, just to show you the great team that actually worked so hard for to the success of that network. So our vision was to advance the application of DNA-based tools for biodiversity assessments, and we wanted to develop a roadmap to include these in standardized monitoring of aquatic ecosystems in Europe and beyond. So we focused on European directives. In a nutshell, right now, what happens, you sample, you process your samples, you identify your sample using taxonomic keys. Based on the taxa list, on comparisons to reference lists, you make an assessment, and depending on whether it's good or less than good, there have to be actions. And the idea that we followed in DNAquanet is complement this approach by extracting DNA or eDNA, create sequences, and based on the sequence list, assign names, and then follow the same way. Or, and you heard the talk by Tristan Cordier, work also with taxonomy-free methods to assess the ecological state. We subdivided the work into groups dealing with reference libraries, index development, bioinformatics, laboratory methods, and the legal issues. And DNAquanet's fear, which was also our motivation, was to not end up in a situation like the traditional system is. And this is, if you look into how Water Framework Directive assessment is being done, then this rests upon a whole tradition of a culture of assessment in Europe. So there are so many different methods because of the individual histories in the countries. There are now, I think, 443 different intercalibrated methods. It was a lot of work for the people doing that. And sometimes you can not really make things compatible, or only to a certain degree. So, and we wanted at this point in time, when the new methods come, to have kind of a top-down control that all people ideally use a very similar set of approaches to not end up with 300 ways or even 3,000 ways to assess Europe's surface water quality, because as comparable as possible is key to success when it comes to biodiversity data. Why are things not being picked up, or why were they not being picked up? At the stage when the DNAquanet started, it was obvious that the technology wasn't ready at all points. There were issues, for example, we cannot generate abundance data, the perception of these technical terms, O2, MO2, ASVs, created frictions between the scientific community and end users, and also the concept, sampling something locally versus filtering water that moves around was quite different. We made a lot of progress with all the people from the different countries and the different sectors, and produced lots of convincing evidence that, and these figures are just some randomly selected presentations by DNAquanet members, if you compare DNA-based to, if you compare DNA-based on the y-axis typically to morphological on the x-axis, usually there's a good correlation. Sometimes you have to transform or arrange a couple of things, but typically there's a good correspondence of the different methods, and this is really good. Also, this is a result of Valentin Vasselon from Schema Bio here coordinated a ring test. And what you see here are reference labs and various labs using different approaches. So many aspects were kept similar, but then various things were varied. And what you see is different labs doing the diatom test come to very similar conclusions. That's very convincing. We showed that you can scale up the process. So this is an example from my lab. We just processed a couple of thousand insect samples within rather few weeks and at very little cost by using robotics, automated robotics for that. So I think that's nothing new to you. And also people in DNAquanet, including us, sorry for taking an example from my lab here, we developed software which is targeted for to the non-bioinformaticians, so graphical user interface software that allow people that want to use the data for biological interpretations can actually work and interact with that. So that also came out of that, lots of research software for sure as well, but bridging the science policy gap and application gap, that was a task of major importance. DNAquanet's progress also resulted in various international and national pioneer studies like the ScanDNA Net project, a validation study of the Nordic countries, or the big joint Danube survey, 15 countries involved in monitoring the ecological state of the Danube, just published, or in the generation of a validation scale for single species assessments. So thanks to those people here shown on the right hand and many more. Yeah, that's a nice DNAquanet result together with the ICPDR, the International Commission for the Protection of the Danube, the publication of macroinvertebrate and fish and diatom diversity along the Danube. Didier Pont was responsible for the fish part. Amazing data that was generated. You can download it in the scientific report. And I could continue now. There are over 100 papers published on this. Just to quickly show you a couple of the key outcomes we checked. Are the reference databases ready for purpose? Where yes, where no? How can they be standardized? What are options for the different bioindicators when looking to the Water Framework Directive? What different ecogenomic indices exist? What are the problems with the novel ones? And how can we revise reference databases, in particular here for the marine realm? Another good product of DNAquanet was the launch of a new scholarly journal, an open access journal that is now, yeah, it's for free open access publication until the end of January. Yeah, you can look it up, but it's well received by the community now. The scores, the official scores, although I really don't care so much about them, they shoot up quickly thanks to all the great publications. But maybe more importantly, beyond research, we try to condense all the consensus we had into guidance documents. And the most recent one just emerged, a practical guide to DNA-based methods for biodiversity assessment, led by Kat Bruce and Christy, who will speak today as well. yes, and many other authors. We disagreed on so many things, but we identified there's still a lot of consensus, and this is what we try to get into this guidance document. It's probably not perfect, but it's the best we managed to get out, and I think it's a fantastic product. We also did this on national levels, for example, for Switzerland, in order to support end users how to make use of the techniques when it comes to practical bioassessment and monitoring. And now the big but. So that all I said was a big success, but my big wish, or our big wish, was to develop a roadmap and actually to implement it. And the idea was, and I guess sometimes we're just too simplistic in our views how the world works. So the idea was we have this amazing scientific network with so many experts also from the applied sector, from the regulatory side, that we managed to install a formal structure. When it comes to the legal frameworks, there are typically working groups in charge of the implementation. When it comes to Water Framework Directive, there's this common implementation strategy from the European Commission, and EcoStat is the main working group that deals with the installation. And our hope was to kind of manage to get, for the mandate period starting 2021, a working group that deals with aspects of implementing DNA and eDNA-based methods. But this was discussed, and I think also EcoStat is very interested in these topics, but yeah, it has to come from bottom up. So the countries have to ask and lobby for it. There was no top-down decision by EcoStat to install such a mandate group, but a lot of interest in this. So that means we have nothing at the top-down level. What we have is, though, lots of national working groups that deal with these aspects in the implementation issues. And what we have is at CEN, the European Standardization Committee, new working group, CEN, Technical Committee 230, Working Group 28, that discusses how to make standards, minimum standards for the uptake of DNA-based methods. However, it's a bit pity that we don't have this top-down control here, and also DNAquanet ended the formal network period. There's no extension at the moment of the formal network. However, if we look at all these national and sometimes transnational activities, I have a lot of hope. If we, and this is by far not complete, but if we look at all the activities going on at the moment with respect to using DNA and eDNA-based data and developing roadmaps, then we see that various countries in the Netherlands, in Switzerland, in Finland, Germany, here in the UK, in Australia, in Canada, in Japan, we have the eDNA Society. In many countries, there are initiatives that at the moment evaluate the potential of the methods and sometimes have rather clear roadmaps and even funding to include these and to complement the monitoring. This is good, and there are also some international activities like Bioscan Europe is currently launching, Europabon, some Interrex where this plays a role. But I think the main strength rests in the national activities where there's permanent working groups and funding for that. So my big hope is that now, rather than the top-down control, we can continue working with this bottom-up control. That means, like the UK DNA working group, work at the national level, but bring in the expertise from as many as possible other countries in order to not develop, to not have a national solo, because what you want to have is kind of an agreed approach so that the data can be used in a by far a larger context than just the national monitoring needs. So I think I'm really hopeful seeing that the national networks do such a great job in being inclusive by including other countries. And we see this not only here in Europe, but also if we look on the other continents, a lot of good progress is being made. And I really like this special issue in environmental DNA where they specifically address the standardization needs. So to sum up, unfortunately, there's no formal pan-European or international working group that formally addresses the uptake of eDNA-based methods into international directives. But since the installation of the Aquanet, several national working groups have been established, and they develop roadmaps for validation and inclusion into the legal frameworks, monitoring frameworks, and most of them are extremely open and international. By the way, there's just the next Finland eDNA roadmap workshop, January 28th. Amazing effort. I really like how Finland also approaches that. That's just the next date for such a workshop on my agenda after this meeting. So to the future of DNAquanet, it formally ended, but we keep the network as a loose network together in order to have an opportunity to exchange the activities, to synchronize. So if you want to join, we will launch every first Thursday of the month at 3:30 CET, so 3:30 p.m. Just a small agenda, and we will also continue with the international conference structure we initiated. We had the first last year. It was an amazing success. You see here on the right-hand side some of the virtual interactions, over 1,000 participants. Yeah, let's see if we can have it in a hybrid format or not. But if you're interested in the final achievement, you can download the report of cost action. Otherwise, yeah, happy to be here and happy to answer questions. Thanks.
Well, thank you very much, Florian. I know that, well, I can't imagine there'll be many people in the audience that isn't aware of DNAquanet, and obviously it's been acknowledged amongst not only the DNA working group right from the inception, but obviously, you know, throughout all the participants, and it's obviously been a huge success. One thing that I did not cover in my introductions, but hopefully people do understand, is that they can ask questions. And so, are you there, Kitty? I can't see any questions as yet, but I don't know if Kitty can see anything that I cannot.
No questions.
Okay, well, in the absence of any questions just now, I'll just ask a question to Florian. I think that's a really good summary, Florian, in that there seems to be, you know, lots and lots of activity. You paraphrase Mike's Louis' comment actually about various eDNA applications don't need to be demonstrated, but there's obviously lots of those national activities. So how do we join even more to create that international entity and move things forwards?
I have no clue. So no, what has to happen is, I think as soon as in these international committees in charge of the directives, the power is accepted, then they will probably establish such a mandate group, such working groups, and start with the uptake. But as a scientist, I think I cannot do more than what we did. We can be open, we can demonstrate it, but now it's really up to the people that have the power to install it. And I mean, we talk to very many, and I think they are open, but maybe it just takes a little more time, and I'm impatient. But yeah, there are clear formal structures in terms of adopting new methods for legal bioassessment, and this is not on us. So we have to convince them to talk to us and include these into these structures. Okay.
Well, hopefully Florian is going to be joining us later for the broader Q&A session. But for now, I would like to pass the screen over to, after thanking Florian, pass the screen over to Christy. And just while Christy's getting ready, I will just introduce Christy. And so, welcome to Christy Diner. Obviously, a great colleague and old friend of the DNA working group. And so, for those of you who don't know Christy, she currently holds an employment as an assistant prof in actually environmental DNA at the ETH in Zurich. Christy did a PhD in ecology in UC Davis, and central to her research is understanding causes of biodiversity loss and redistribution for ecosystem functioning. And so she's got broad experience in the field of molecular and community ecology. And she's also the co-founder of the company Simplex DNA, which is currently ramping up in its first stages. And hopefully many of us will know of her seminal work focusing on conveyor belts of biodiversity in riverine ecosystems. And today she is going to be, well, she's going to be sharing, you're going to be sharing your work on global biodiversity monitoring. You're going to be sharing other work, Christy, because I know that you changed the title.
I know I pulled a fast one on you. Yeah, I just got inspired by listening to all the amazing week we've had together and thinking, and so I thought I would shift a little bit and highlight some of the things that we are working on that people have been asking a lot about. So I shifted a little bit, but I will cover our global work we're doing on lakes. So
Great
good.
work. Well, over to you, Christy, and we'll look forward to hearing your talk.
Yeah. Okay, well, thank you so much for that lovely introduction, and I hope you see the right screen. I think it's good. And so I changed my title to "From So Simple a Molecule Endless Forms, Most Beautiful Are Monitored." And if you're a fan of Darwin, you'll know where that comes from. And I guess I'll start with why maybe Florian and many of us feel very impatient right now with why we can't integrate and utilize this tool to its maximum benefit. In this crowd where many of us are quite convinced about the applications and what we can do with this, we stare at other graphs like this and realize, there's these statistics out there like 1 million species at risk of extinction. And I was recently thinking about the gravity of this and what it means. And if you look at the animals pictured on there, these estimates and extrapolations mostly come from vertebrates. They mostly come from maybe a little bit more broadly eukaryotes. And if we compare that to how many eukaryotes we think are actually on the planet, this is kind of one of our best estimates. So 8.7 million species plus or minus a million. And when you do the math, 1 in 8 eukaryotic species are at risk of extinction. Let that sink in. Think about your favorite 8 species and now imagine one of them gone. And, that makes me very impatient. So, but there's hope. The world is really mobilizing right now in a way that I haven't seen in the last 10 years. You see headlines like this pouring out day after day. Money is now flowing into conservation. It's flowing into this problem. We have a huge opportunity right now to harness all of this financial incentive to go out and fix the world, right? And we talk about bending the curve and changing that trend, and that's amazing. And what we have staring in front of us is, this ultimate weapon, if you want, our data solution. And so today I'm going to go through kind of like a little expose of all the stuff we're working on and really try to hit home that we can do total biosphere monitoring with eDNA. And I'll talk about the stuff we're doing at a global scale with looking for hotspots of eDNA and lakes. And I'll talk about air, which is everybody's really excited about now. This is work we're just getting underway. And then I'm also going to talk about how we're trying to integrate both air, water, and soil sampling to get at ecosystem level and biosphere monitoring across Switzerland. Okay, so to dive in, I'm gonna just highlight first all the work we're doing in water. And this is my great crew of both collaborators, students, and postdocs that are working with me on this ERC research funded project. And as you know, this is building quite extensively off this idea that was one of some of the first data sets that I was looking at with eDNA, which was, we found that it was being transported in rivers. And so if you think about what's going on with eDNA, it's basically a function of being moved around in the water cycle. And this is pretty, sort of where is DNA in any of this? And I think we're at the tip of the iceberg of knowing if fog has DNA or, if it's evaporating and all these kinds of things. But one thing we do know is that it's showing up in the surface water, right? And it's running off, and it often goes to a lake or to water or to the ocean. And so now I've been working on this hypothesis for a while now, which is that, basically are lakes forming these eDNA hotspots in a landscape through the transport of DNA off the landscape, both terrestrial species, aquatic species, and then that gets down to this lake and it holds on to it for a short period of time. And this means that transport potential, the movement, has to exceed the degradation. So I'm going to spend a little bit of time talking about how we can think about transport, and then also how we're looking at trying to really get a handle on how DNA is degrading. Okay, so why this is profound is that, yeah, lakes and their watersheds cover a decent percent of the earth. So this is about 25% of the land mass. And many people have asked, how many lakes is that? It's about 1.4 million lakes. Maybe that's doable if we think about all the sites we could potentially monitor with eDNA. However, about 1 million of them are in Canada, so maybe we don't need to sample so many lakes in Canada. But we can spread them out throughout the world, and we might be able to get really a good access to a pulse on biodiversity if DNA is getting to these places. Okay, so this is some recent modeling work that we've done. And if we just make one assumption, which is that DNA is moving at the rate of the water, so the discharge of the water from its watershed. So you can imagine the farthest point away in a lake watershed, so that upper headwaters of a river, and how long it'll take for the water, once it's in the river channel, to get from there down to the lake where it inflows. And we've done a global estimate of that. So all these points here that you see are the estimate of time it takes for the maximum distance in that lake's watershed to get to the outflow. And this is in days over here. Sorry if it's a little bit small, but we're looking at somewhere between some hours to a couple days for most lakes in the world. Okay, well, that sounds like that's not that much time. Maybe DNA really is getting to these lakes. And so now we're launching very soon. I know you guys have been hearing me say this for a long time, but we're going to be seasonally sampling a set of lakes throughout the world, and I'm still building that network of people who are interested. So if you would like to get in on the I guess what Ryan might call the game show of doing some exciting work to test this across the globe, please let me know. And we've been doing some pretty exciting innovation in our lab, and if you've seen me talk about this before, I usually had about 100, 120 lakes on there, but we think now through innovation and both the methods, like Florian was talking about, upscaling with robotics and making stuff a lot cheaper, as well as some really new novel sampling methods that we've been working on, and we hope to roll those out quite soon. So maybe we'll even be able to do 1,000 lakes seasonally over the next year. Okay, so there's lots more to come on that, and we really need to test this, and we need to test it at a global scale because we need to understand if it works just in some places or everywhere, and then could we really build up a global monitoring system based on this idea that we have eDNA hotspots out there. Okay, so now I'm going to switch and talk about the other part of that hypothesis, which is this degradation piece. Now, DNA is this fundamental molecule to life, so it's no surprise that it's also in demand by life. All the components of DNA are used by life, and they're part of the carbon. It's part of the carbon cycle, the nitrogen cycle, and the phosphorus cycle, and therefore, we think that the major control on this is microbial communities through the demand of these particular molecules. Now it's sort of getting recycled, if you will. Maybe it's our best recycled molecule, I don't know. But anyway, so it's getting out there and stuff's breaking it down. But there's also other things that might be doing that, and I'll get to that in a minute. And but when we do experiments, this is a snapshot of the various experiments that have been done, and we see how long it's out there in the environment. It looks like it's somewhere between hours and weeks. And what I've done here is we've taken all the sort of K. So this is a constant that's estimated when you fit an exponential curve, decay curve to the disappearance of your DNA. And then what we've done is said, okay, well, here's the K we got from those studies, and we assume everybody starts at the same copy number, which is always why it's super hard to compare between studies. But if we use this model to do that, we can model how those degradation rates look. And if you go here, this is time in hours, we're in about 2 days. So if you remember, I showed you water moving to lakes is in that range. So we're probably still seeing DNA get there and potentially accumulating. Now, what are some of these other factors that may be driving that? So this is a recent meta-analysis that a few of us just conducted. And so we went out and we took, this is just for fish, it's the most well-studied group out there. And we looked at how temperature drives degradation. So each of these points is a measure of degradation of DNA from an experiment on fish. And we put this into a special kind of graph, which is based on the Arrhenius equation. And this is just that you see activation energy, so the enzymes maybe driving the breakdown of DNA is influenced by temperature. So a specific unit of temperature leads to a certain activity, and that drops or increases the degradation rate. So this is 1 over T, which is in Kelvin actually, and here's the rate, those decay constants we are looking at, and the points are color coded by temperature, and sure enough, the colder it is, the slower the degradation rate, the higher it is. And so this is really exciting, but you can also see the R squared, maybe most ecologists were kind of used to going, yeah, that's cool, we got it, but there's still a lot that's unexplained. And so there's still a lot of factors that we need to understand what's going into this, and we're really trying to dive into, well, does the state of the DNA matter? Does that actually play a role in how long it persists? And is it different between plants and animals? What happens if it gets bound to different types of particles? Does it get protected forever or not? And, of course, these other abiotic factors, and really trying to quantify the effect of bacteria. So we're diving into doing a lot of these experiments, and I'm not going to go into too much of that, but I'm going to talk about why this might be important. And this gets a little bit into the ecological interpretation of time and space. So when we find that DNA molecule in the water, how far did it travel, and how long has it been there? Those are big questions that we need to rein in. And so what's interesting about states is if they do have different decay rates, then we might be able to rein in the spatial and temporal scale in the inference we can make if we focus only on one. And so, based on what we understand or hypothesize how it works, we might be able to attribute different spatial and temporal resolutions to these. And so then the question becomes, can we actually separate those and study them individually? And this is exciting work that Anish in my lab has been investigating. So we designed this very elaborate experiment and used DNA that is it's different species, but the species were introjected in the experiment in different states. So we had adsorbed DNA onto clay, and this was only salmon DNA, and free DNA, which we extracted from chicken breasts from the store, only chicken, and we had membrane-bound, so cells from mouse cell lines, and we put them all in, and we said, okay, well, if we use our molecular, toolbox, and we say, okay, this is how we think we can separate this different states, how good are we if we were to have a liter of water, if we run it through this pipeline, could we spit out only chicken DNA, mouse DNA, and salmon DNA based on knowing their state? And we tested this a couple different water types, so that's what you see here with the different colors. But the important thing is that we have DNA copy number here. So for the membrane-bound state, this was our expected state given the methods we applied to this. We found mostly membrane-bound DNA states, but in those other pools of DNA that we created, we still detected some of the mouse DNA when we did not expect that. However, if you look at the x-axis here, this is an order of magnitude different. So we're doing pretty good. And if we can start to understand the relative biases and the efficiencies of these extractions, we might be able to really hone in and study specific states. And that might get rid of some of this inherent uncertainty and biases that we might have, because until now we don't really focus on any one of these per se. Okay, so that's really exciting. Now I'm going to pull us out of the water and get our head back in the air, because I only have a few more minutes left, and I just want to tell you about this really exciting research that we've started, and this is being led by Fabien Roger in our lab, and he's got this Mary Curry grant fellowship to come here, and he's just getting off the ground, and yeah, he wrote to me a while ago and was like, yeah, I have these really great ideas for doing this. And then of course, all these papers have been coming out. And so people are interested to fund this, and it's really exciting. So if you have ideas to advance this, get out there and try and get some money right now. Everybody's everybody's excited. So what are we going to do? Well, so there's, obviously the few papers that just come out, everybody kind of did something different. And we're going to look at comparing some different methods. So there's kind of a high-end vacuum that sucks air into the water and in a vortexing thing. But there's also these commercial vacuums that do the same thing. So we're going to test this on this amazing grid system where biodiversity is monitored in Switzerland. And the other exciting thing, and this is something that Fabian came up with, which is that we monitor air for pollution, and there's these stations all over Switzerland, all over Europe, all over the world, and they actually filter air for 24 hours. So they take a pretty large volume sample of air, and this infrastructure is already out there. And so what we're going to be looking at is whether or not we can actually get DNA off of these filters. And for those of you who've been working in eDNA a long time, not all the stations use this, but guess what fiber, or guess what material that filter is made of? Glass fiber. So we have a pretty good idea how to get DNA off of that. And if this is working, then maybe we already have an up and running air monitoring system all over the world. Wouldn't that be incredible? Okay, stay tuned for that. And then unlike, very similar to our work in water, we really want to contextualize and understand what are the particle sizes, how do they move, can we predict where those air masses are coming and going, and really try to get in on this. So we're starting in the lab, trying to figure this out by putting insects and birds and stuff in these aerosol chambers and trying to see where the signal's coming from. So stay tuned. This is all getting underway and we're really excited about it. So now I'm going to talk about the soil. And this actually was started because we thought, okay, we're going to go out and look in the terrestrial environment and see what we can do. But then we thought, well, why don't we go ahead and take water and air too? And then we asked, well, can we monitor the entire biosphere with just these 3 samples? And would that get us towards something of understanding function? So we went out and we did all these and we took all these samples and we're just in the middle of processing that. So here's your very first slide. So we started with bacteria. Bacteria are kind of an easy go-to protocol. It's so well understood how it works. And this is an NMDS plot. So the farther away these points are from each other, the communities are more dissimilar. And we see this pattern that we get different diversity between water, soil, and air. So that's interesting. These samples do tell us something that's more than just taking one of them. But when we look at the shared ASVs across all of this, sure enough, across all these 44 sites, we even get, enough ASVs that are shared across all of them. We see stuff that's only shared between water and soil and air and water. So this is telling us that there is complementary information as well as things that are the same. So we can potentially turn this into functional aspects. And now we're looking into what taxa these are and does it make sense that they're shared across all these different parts of the biosphere, so on and so forth. Okay, so hopefully I've gotten you excited and teased you a little bit about all this, and really this is just to mobilize everybody to be thinking, we don't have to limit ourselves just to water eDNA or just to soil or just to air. We can be thinking about these and doing all of them. And the methods are out there now. We have a lot to go, but way to go. Okay, so now I'm going to throw in really quick, very last little point. Everybody was asking about population genetics data, and I'm going to tell you a fun story. And the punchline is, yes, we can do PopGen with eDNA. So we went to this iconic place. This is Lake Tanganyika. And we went here because there's a very iconic diversity of fish, the cichlids, and they have this cool evolutionary history in the lake. So about, during the end of the Pleistocene, the lake was dried up and there was these 3 proto-lakes, and this actually led to a lot of isolation between the lineages of cichlids. And today that lake looks like this, and we went out and sampled in 2 areas that represent these kind of 2 proto-lakes. And the number of species we're talking about within a family of fish is represented by these different colors here. So depending on where you are, you kind of get a different estimate, but somewhere around 100 species. Okay, what did we find? Okay, so yeah. Here's those proto-lakes, here's our sampling locations, here's the haplotype network that we're recovering from one of the species that's a well-known species to have this very north-south divide. So these are haplotypes recovered from water samples. Now, we did this simultaneously for over 100 species. Maybe some of you saw me tweet this the other day. I mean, it's just, yeah, you can't make this stuff up. It's pretty crazy, but this is just for 160 liters of water. So, you know, it's about using the right gene. The barcoding genes we use for species level are not the genes we should be using for meta barcoding or meta population dynamics. You have to go to different genes, and that's what we did here. But the information's in there. It's all about using the right tools to get access to that information. So I just wanted to do that, and I'm going to do one last thing, which is tell everybody that we're launching a special issue in environmental DNA. This is something that didn't come up too much this week, but really trying to advance disease ecology through co-detection of pathogens and their hosts, and how eDNA can play a role in infectious agents and monitoring. So we have a really great panel of guest editors that have joined us from USAID, EcoHealth Alliance, and researchers who are deep in studying pathogens and parasites. So please, if you're interested, reach out and let us know. And with that, I will just say we're open for business. So if you have any eDNA needs, let us know. Thank you.
Thank you so much for that, Christy. Great to see how your lab is diversifying across all these different threads, and obviously all the best for your ERC project. I know that everyone's really excited about that. And so just to remind everyone, including the speakers, if we find we can't get through all of the questions, Kitty will be assigning questions to the speakers. So just glancing back to Florian, I think Florian has also dived in there, and we have, we've got a couple more questions coming in, so I'll pass over to Kitty.
Okay, the first question asks, do you think the variability in decay rates among taxa is mainly driven by the organisms themselves? That is, different DNA shedding mechanisms resulting in different eDNA states, or by different environmental conditions from the different studies, or were these somehow standardized?
That's great questions. I would say we don't know. I think there's good evidence for both the species and the environment playing a role. And I really think it's fundamental to study things that are like the cell type differences, so that's why I think plants and animals might behave really different, because a cell membrane and a cell wall are fundamentally just different, and how those behave once they get released into the environment, then they're definitely interacting there. And how much shedding is happening, so that amount that gets in there initially and how it breaks down into different states is, yeah, there's a lot to understand about the whole cycling of DNA in the landscape, and we need as many studies as we can do on that. So we'll never get tired of them. Submit them to eDNA. We'll publish them, because we need all that to build up that space that we can see, do meta-analysis and really see which one explains better. But right, those things are coming out, so we'll learn a lot in the next few years, I'm sure.
Okay, if we just have one more question, please, Kitty.
What groups are you looking to identify from your air sampling?
For the air sampling, yeah, so we're sort of throwing the whole war chest at it, the tree of life approach. So we're really looking across the all three domains of life. So we showed you the bacteria, but we'll be looking and seeing how we can do everything. Why not?
Great stuff. Well, we'll look forward to hearing all about that in the future, Christy. So thank you so much. And so what I'm going to do now is I'm going to switch over to Nayara. There's so many panelists today, which is good. And just while Nayara is enabling sharing her screen, I'll just provide an introduction. So Nayara Rodriguez Espeleta, I hope I've said that correctly, is a senior researcher at ASTI, a marine and food research focused tech center in the north of Spain. So there she leads the eco-evolutionary genomics applied to marine management research team. And her work focuses on applying genetic techniques to provide useful information for fisheries management and marine monitoring. She's currently the chair of the ICES working group on the application of genetics for fisheries and aquaculture, and has previously chaired the ICES working group on the integration of morphological and morphological and molecular taxonomy. And so Naira has been very busy. She's published over 70 scientific articles and book chapters, and she's an editor. Oh, well, an editor also of molecular ecology and molecular ecology resources. So great work, Naira, and we look forward to your talk.
Okay, so can you confirm you can see my screen? Everything is correct?
All good. Yep, yeah, all good.
Okay, yeah, so I'm going to talk to you today about the work we've been doing on estuaries, on the pelagic realm, and on the deep sea. It's a little bit difficult to make this talk interesting and to tell something new after all these amazing talks these days. So I have to admit I changed a little bit just today. I removed a lot of material that I think it has already been covered. So yeah, I can perhaps say something new and hopefully interesting. So yeah, I told you I'm going to be talking to you about these 3 cases, and actually they are at different levels on how ready they are to be used for management. There was this discussion the other day about how we should make eDNA boring, meaning useful for management as it is now, and keep also doing research to understand it better and improve the methods, the protocols, and so on. So when I'm talking about how ready they are to be applied for management, I'm not saying how ready we are to transfer the results, but how close the potential or hypothetical results we would get would be from the management needs. Let me explain what I mean. For example, in the case of estuaries, we are working in the framework of the Water Framework Directive, as Florian explained today, and also was mentioned in some of the talks before. Actually, in this case, we are looking for indices that tell us about the ecological status of an environment, in this case of estuaries. So in this case, the need from the managers is clear. It's really the index that we need to provide with the eDNA data. That's why I mean it's close to be management relevant. In the case of the epipelagic realm, we have a framework. So we have a management framework, which is the Common Fisheries Policy, but it's less clear what the managers need from us, what type of data they need, right? So obviously, we want to have fish biodiversity data. We want to know about the abundances of the different fish communities and so on, but it's not so clear what is the data, what is the number, what is the output they need from us. But we have a management framework. And in the case of the deep ocean, we are a little bit farther from management because we are still in the process of gathering scientific knowledge. There is so little known about the deep ocean that we are still learning about it, about the role of the deep ocean, so that we can then establish the needs from the managers in terms of scientific knowledge needed, in terms of in order to make useful and efficient management actions. But before I go deeper into from the estuaries to deep ocean, I'm gonna go out a little bit and talk about the elephants in the room of eDNA. This is things that we all have in mind, all working in the eDNA, we all have in mind all the time, I'm sure. In red, I put the things that I'm more concerned about now. So there is work from Christine on this index hopping, sample bleeding, and so on that I've been following, and I think people are confident now that we can solve it. But I'm still a little bit concerned about it. Same thing with controls and so on. So anyway, I leave this here for in case this is useful for the Q&A session. But I'm going to talk to you now a little bit more about database completeness and accuracy. This is something I wasn't planning to include, but I thought it could be interesting to have it as material for discussion. So we've been reviewing the papers using eDNA metabarcoding in fish, and actually we have realized that most of them rely on GenBank for their reference database. What happens is that in some cases, there are some tools that are there to make our job easier and to help us build the databases with the markers we are interested in. So I'm putting here 2 examples. The problem with these databases is that although they contain the markers we are interested in, they carry out the mistakes that are present in GenBank. So if GenBank has incorrect records, they will be present in those databases. So those would be taxonomically complete, but they would carry out mistakes. Another approach is to extract relevant markers, but also taxa. For example, here is an example of a database of UK fish species, right? So you extract the species you are interested in. So you have a reduced set of species, and then the good thing about this is that you can curate the data set because it's small enough, so you can make phylogenetic trees and you can check for inconsistencies and so on. But they will contain a reduced number of taxa, so they are not adequate for global analysis. In our case, for our needs, basically, for the estuaries, we are working on a quite restrict area, so we are working quite locally, so we could actually try to build our own database. However, for our epipelagic and baripelagic work, we are working on different areas across the Atlantic and Mediterranean Ocean, so we need a quite extensive database. So basically, our aim is to create a database that covers at least this area here, so the Northeast Atlantic and adjacencies. So this is what my former master's student and now PhD student, Cristina Claver, has been working on. So she's been trying to generate or to check actually how complete and accurate is GenBank for fish eDNA metabarcoding in the Northeast Atlantic, and to try to see if we can come up with some kind of automatic way to make this database more reliable. So, well, she collected the species that were present in the Northeast Atlantic, and then she checked for completeness and for accuracy of the database. So she did this for the 4 most commonly used genes for eDNA metabarcoding of fish, so CO1, 12S, 16S, and cytochrome B. So here is the coverage for each of those genes. So this means the number of species, the percentage of species from the ones we identified that have that gene sequenced more than 10 times, from 2 to 10 times, or 1 time. And this is what it shows. So the CO1 is the one that has most coverage. We can also do this per group. So being Actinopterygii and Elasmobranchi, the most abundant groups, and more or less they have the same coverage for all the markers. And concerning the barcodes, so for each marker, we check the most commonly used barcode. So here would be the long or short CO1 barcode, universal barcode, or the Tilio or MyFish regions. So we checked also the overlap, actually. So in the case of CO1, both barcodes have more or less the same coverage. In the case of 12S, Tilio has a bit more coverage. It's a shorter region. It has been sequenced in more species. So why don't we use CO1 for fish eDNA metabarcoding? Well, the reason is that at least the most commonly used barcodes for CO1 amplify also other taxonomic groups, not only fish. So even in chordates, we can find in our water samples about 2%, a bit more than 2% of the sequences are chordates, so very little. And even though most of them are fish, actually we cannot really we don't have a very good taxonomic resolution. So basically we cannot really use CO1, at least with those primers, for eDNA metabarcoding of fish. So focusing on the 12S barcodes, we wanted to quantify somehow how accurate is GenBank, like raw GenBank, without doing any type of filtering for these barcodes. So here what we did is that we did a blast against all against all blast, and we calculated the distances between sequences, all the sequences we downloaded from GenBank, and we calculated, we separated into those sequences that belong to the same species, those sequences that belong to the same genus and not the same species, and so on. So you can see that, as expected, the sequences that belong to the same species are more similar to each other, and as we get further in the taxonomy, they differ more. However, we have some suspicious points here, right? So we have sequences that belong to the same species, but they are very different to each other. So those could be potential errors. Or could be species that have a very high internal variability, but this could be a little bit, I mean, it could be some cases, but obviously this is not the case for most of them. And then we have other cases where we have sequences that belong to different orders, to different families, different classes, but that they are almost identical. And those are potentially contaminations. So we tried to put some numbers into this point. So we actually quantified how many pairs of sequences fall into the different categories, so species with species and so on. And we identified that the problematics would be those that are taxonomically apart, but they are very similar, and those that belong to the same species, but that they are very different. So there are some cases that we actually can explain. For example, this is one of them. So we have a case where we have two sequences that belong to a different genus, but they actually, one of the genus is considered the other one. So both, in this case, Boreogadus would also be considered Gadus. So those are examples that are quite difficult to handle automatically, because we could label this as a contamination where actually there is not. So it's not so simple actually to clean the database. So we came up with an idea of trying to do some networks and to see if we could use those networks to semi-automatically filter some potentially erroneous sequences. So for example, if we go here are the networks showing groups of sequences that are almost identical to each other, but that they belong to different genus or different class, different order, and so on. And here is one of the examples we found. For example, this is clearly a contamination because those are species from different order and they are almost identical, right? So this could be an example of one of these type of problematic sequences. This is work in progress. I don't have the final results to show you, but I wanted to put it here as something I think that hasn't been discussed in detail yet. So I think as a material for the discussion later. So going back to the examples that I was explaining before, so the estuaries, pelagic, and deep ocean, I'm going to show you the results very briefly we have for each of them. For the estuaries case, as I told you, we've been working on the Water Framework Directive framework. And the idea here is to come up with an index that summarizes the fish community, and that this index will be indicative of the ecological status of that estuary. So basically, this index will consider the proportion of individuals of the fish community we sample that are, for example, pollution indicator species, that are introduced species, that are flatfish, omnivorous, etc. So this index has been developed, and we analyze the community, and then we calculate a number. And then this number will summarize the ecological status of the estuary. What happens is that usually the way this is done is that we use trawling, and we collect the fish that appear in the net. The problem is that we have our estuaries in the Basque Country are really, really small, and sometimes they get it even empty. So we can only do this at high or rising tide. We can only do that at soft bottom. The noise generated by the boat and also the transparency of the water are known to affect the catches. So we don't get the same type of catches when the water is turbid or is more transparent. And also we don't cover the full range of species and locations because it depends where we can go with the boat and also the mesh size and all those kinds of things. So the idea is that with the eDNA, we could overcome these issues and have a better view of the fish community composition of the estuaries. So we sampled 12 estuaries, 12 estuaries in the Basque Country. We sampled them the inner part, the middle part, and the outer part of the estuary. And then we collected the fish with the net and also the eDNA. We sampled 2 liters of water and we amplified the teleo region. So here you can see the number of species that coincide between the trawling and the eDNA. Very little. In eDNA, we get many more species, but also we had to put this in the context of the database. So there are some species that are detected with trawling, for example, and not with eDNA, but they are not present in the database. So it's normal that we don't detect them with eDNA. And then we put them in context in the species used to calculate the index, which is called the AFI index. So when we take into account only the species included in the AFI index, what we find is that this index is usually lower when we use bottom trawling and higher when we use eDNA. So we are calculating better ecological status of estuaries if we use eDNA than if we use bottom trawling. Even when we use only the species that usually appear in the trawling, because we thought maybe we are capturing with the eDNA species that never appear in the trawling. But even if we take only the species that appear in the trawling, using the list we have through the years, we can see that actually we have always better ecological status with eDNA. If we separate the different parameters considered in the calculation of the index, you can see that the richness is a big difference between the eDNA and the bottom trawling. So the richness is always going to be higher with the eDNA. So what's going on here? So the message here finally is that we cannot just directly apply the index used with the fish to the index, to the samples, the eDNA sample. So we need to adapt somehow. It's not a direct thing. So we are here close to management in the sense that we know exactly what we want to get, but we don't know how to do, how to deal with the differences that we obtain with eDNA and the in the trawling. Okay, so in the case of the epipelagic realm, so this is work by former postdoc Natalia Franja Fernandez. Our idea was to provide data that could be used by the common fisheries policy. So we wanted to assess fish diversity and biomass. So this is traditionally not done with the scientific surveys. Scientific surveys that in our case, in the Bay of Biscay at ASTI, we've been doing our survey since 30 years. So it's been going on for 30 years. And since 2017, we started collecting also water samples for eDNA analysis. So the idea is that the eDNA would provide us good view of the community composition of the fish in the Bay of Biscay, and that it could be an alternative to the scientific surveys, which are quite costly, selective, and able to detect rare and elusive species, and they are also habitat destructive. So could eDNA be used to reduce these problems that I highlighted here? So we sampled in the Bay of Biscay. So here the crosses represent the catches, and the black squares, circles, and triangles represent the eDNA samples. So, and we compared what we obtained with the catches and what we obtained with eDNA. As usual, the number of species collected by eDNA was much higher. So here there are more than 83 more than the ones listed here. But the common ones were the same. So the really abundant ones with both methods were the same. The interesting thing is that there was correlation with the eDNA number of reads and biomass from the catches. And what I think was most interesting from this study is that we saw a vertical structuring of the eDNA. So the eDNA from the species that are usually living in deeper waters increased as we move far from the coast. And the eDNA from those species, the abundance in eDNA of those species that are living in shallower waters and closer to the coast decrease as we move far from the coast. So this was quite interesting to see in this open ocean, to see this very clear differentiation in the verticals, in the horizontal scale. And the last one, and this is very short, exploring the dark ocean. So we really wanted to see if the eDNA could provide information to understand more about the dark ocean. The dark ocean and the deep ocean is seen now as a new opportunity, as a new source of food for humans, or as food for aquaculture, or as a new source of new compounds. The problem is that the deep ocean is also important because it has a key role in trophic connectivity and carbon sequestration. And this is because of the diel vertical migration that some fish do during the day and night. So some fish and other species as well, what happens is that they go up during the night to feed, and they go down during the day to escape predation. So we wanted to see if eDNA was able to see and to reveal those patterns. So we sample again in the Bay of Biscay at different depths. This is the list of deep sea species that we found. The most abundant one in the non-deep sea was the anchovy, and the most abundant in the deep sea species was the Morolicus molleri and the Lophius piscatorius. As you can see, the abundance and riches of deep sea species increase as we move down, as we go deeper. But what is more interesting is that we see the diel vertical migration pattern in the eDNA as well. So we see that during the day, the eDNA from the deep sea species is found in deeper waters, but during the night, the abundance, eDNA abundance of the deep sea species goes up. This is at the community level, but we can see this as well at the species-specific level. So here is an example of 3 species, 2 perform vertical migration. So we can see that the eDNA during the night increases at shallower depths. However, there are some species that don't do those vertical migrations. They are deep sea species, and their DNA is always found in deeper waters. It is interesting here that we also have some sinking of DNA, probably attached to particles or something, because we find always DNA even of the only surface species in deeper waters. For example, we find eDNA from anchovy in deeper waters. So finally, just a little bit of food for thought here. In the case of estuaries, high detection potential of eDNA needs to be accounted for when developing eDNA-based index, but how should we do this? So we are working on this. eDNA is powerful to assess fish diversity and abundance and could substitute or at least complement scientific surveys, but what type of data should we provide or store? How do we deal with historical data? How do we deal with time series, for example? And finally, eDNA is a powerful tool to explore and understand the dark ocean. And the question now is, how can we get to know about the rare, least abundant, right? There are very few abundant species that sometimes we don't catch with eDNA. So we really need to work some protocols and methods to be able to catch the eDNA from this least abundant species. And that's it. Thanks for listening and for the invitation, and happy to answer any questions if there is time.
Thank you so much, Nayara. It's really great to see those results coming from the marine environment. And I know that Louis was definitely covering some of those experiments before. It's really interesting that you can see that diel vertical migration using the eDNA, and I think it just adds to that fine scale variation that you can pick up with environmental DNA. It's really good to see those results. So Kitty, are you there?
Yes.
Are there any questions for Nayara, please?
Yeah, so there's one question. It says some researchers are suggesting identifying and working on creating a single standard universal marker or reference database, while others are suggesting multi-marker approaches, complementary for more holistic views on community diversity. What are your thoughts on that?
Well, my thought is the easy answer. It depends on the question, right? So, I mean, for me, it's very difficult to make something universal for a variety of questions. We should try to universalize the work we are doing to answer the same question so we can compare. But it doesn't make sense that I use the same marker if I'm going to analyze, I don't know, fish that are very close to each other from, and you want to have a marker that can really distinguish species. Maybe I'm interested more in the community overall, and then I don't need a marker that is able to distinguish species so much. So I guess it depends on the question.
We're more or less on time, and so we're going to move on to just kind of like a wrap session, but then also give the audience an opportunity to have a general question and answer session. And so what I'm going to do is I'm hopefully going to take you back to my screen. And just before I move on with any scientific discussion, I think it's really important just to thank that we thought this would be fairly a low level exercise to organise this webinar week, but it's actually taken quite a lot of effort, and I've been helped by a large group of people. And so I'd personally like to thank Andy Nisbet and Katie Clark, obviously hosting the first day. And then obviously apologies from Stefano Mariani. He was helping throughout, but obviously also his postdoc Wang Kai helped on Tuesday. And then Laurie and Tabitha presented on Wednesday, followed by Alan McDevitt and Holly. I think it's Alan. They did a great job on the industry day. And then obviously also Kitty for supporting today's session as well. We haven't seen the faces, but we may see the faces of Dan Reed from CEH. So he actually enabled us to use the GoToWebinar interface. And even though we were quite worried about it, it's actually worked really, really well. And it's been really powerful to enable us to share screens with so many people with all sorts of different operating systems around the world. So that's been really useful. And then also Phil Davison and also Doug Yu were helpful attending many meetings and assisting with the sort of like scientific guidance of how we devise this webinar series. But I think most of all, I think on behalf of all of the organising committee, we'd like to thank Tabitha Blackwell. We may see Tabitha in the panel, and you definitely met her on Wednesday. She was supporting Laurie. And obviously, well, I know that without Tabitha, this would not have happened, because Tabitha was doing an enormous amount of heavy lifting when it came to communicating with the various speakers and coming up with timetables and dealing with all sorts of logistical issues that we may have actually struggled to get to. So Tabitha, I've sent something that well, I've sent something via Sandy that she'll give you as a small thank you present. So hopefully you'll get together with Sandy to receive that just shortly. And so just moving on, I'm going to just invest a small amount of time, and it is going to be small just to allow time for broader questions, just investing a bit of time reflecting on some of the talks that we have seen. And I don't know, I'm assuming you can see my white screen and you can't see any strange Q&A because from the webinar. But so if anyone from the organising committee or panelists can say whether you can see my screen, that would be good.
We can see you. Yeah.
Okay, good one. Okay. So I'm going to go back to this. Okay, so just briefly, I think my reflections on this week has been, I've noticed a number of senior colleagues all using the word amazing, and that is an adjective which is being used to describe all the research that we've seen this week, and obviously all the quality of the speakers and their presentations. So hopefully the audience have valued their contributions. I think also with this webinar series, even though we normally have physical events, we've also almost seen like a maturation of the UKDNA working group, obviously in collaboration with UK EOF as well. I mean, just the fact that we've now got a logo and we've almost got like a web home to go to on top of all the efforts from the members, I think is absolutely great. And then also just reflecting and looking back at the volume and intensity and clarity and findings from the studies, we can see that the data is really maturing. What people are finding from this field, and also more or less everyone is looking towards scaling their biodiversity assessments up. So we've moved right from the 2008 Ficitola paper looking at the invasive bullfrogs to now many people are using multi-marker approaches and looking across the tree of life and thinking about ecosystem scale assessments. And I did really like Louis' comment, and obviously Florian reflected on this, that he felt now, having devised a new journal to convey the research from the field of environmental DNA analysis, that he doesn't feel that the validity of environmental DNA analysis in aqueous system does not necessarily need to be further demonstrated after his reflections and looking at the papers he presented. But obviously we need to move from these amazing lists of taxa to ecological understanding. So Kat raised this point in her talk. It's good to see all these lists of things, but we need to understand what those things are doing and how they're interacting. And I cannot remember, it was Sarah, and Sarah actually pointed out that she also felt that the terrestrial biosphere needed an analog of DNA Aquanet as well, so that they could harmonize those efforts in the terrestrial biosphere. We were also reminded by lots of speakers that, especially to those colleagues from the existing biomonitoring frameworks, that eDNA is a complementary method. And so what we're doing is providing additional information that may not be possible to achieve using current approaches. But also, as summarized by Kat, because you can generate this data, then it frees up time for traditional taxonomists, taxonomists and field ecologists to do additional work. They can do more assessments, different forms of assessments helped by the environmental DNA data. I also really like the analog by Ryan when asked by Andy about what do agencies do about the evolving technology of environmental DNA research. And I thought Ryan's point about the DNA fingerprinting, which is a fairly standard DNA profiling method, has been used robustly for over 30 years now. So just because things change, it doesn't necessarily mean to say that the approach has to change, definitely not in that field. And then obviously we saw from Melania the power of metatranscriptomics when it comes to understanding stresses and functional indicators. And I was really impressed with the industrial representatives from the different companies. And so it's absolutely fascinating to see that there is now a growing industry focused on the provision of environmental DNA data to different end users that potentially are not governmental end users and stakeholders, but different forms of commercial partners that are interested in this data. And I think, as alluded to by Andy on the first day, we're hoping to release all of these webinars on a YouTube channel. And I'm sure that Dan will be able to let everyone know who is registered how we're going to do that. And I would really recommend that people take note of obviously the speakers' talks and dive into their presentations to look at the key references that they've shared, because these ladies and gentlemen are kind of thought leaders in their field, and if they're talking about those particular papers, then those are the ones that they've identified to get those messages across. And just also, I think, from looking back from the first sort of 2014 UK DNA working group meeting, it's really nice to see the legacy of all of our friends and collaborators in the field, and then also all the postdocs and students that we've seen across the board that are really now becoming the next generation of eDNA scientists. And so that's really my wrap-up session there. And I'm also just keeping my eye on time. And so I think that gives us about 35 minutes or so for the people in the audience who do want to participate in a broader session. Then I think now is the time to try and achieve that. And so if I can ask any of the speakers and indeed the organising committee, if they can turn on their cameras, then we'll see who we've got. Great, I've never seen so many people in the GoToWebinar window. And also just really invite, well, it can be members from the panel, it can be members of the organising committee, or indeed the audience, and hopefully Kitty is going to be able to help to field any questions that we do get from the floor, to see if people have got general questions that people may want to answer, or indeed ask maybe like the general audience, or maybe if you've got specific questions that you couldn't ask during the sessions that you may want to ask specific speakers that are present. And so I'm assuming the audience is still there, and I'm assuming they may have some additional questions. But if they don't, how about the speakers? Have you got any points that you would like to share in a kind of like a summary session? So and yeah, the hand is good, Florian. So first point to Florian, please.
Okay. Yeah, one question. I mean, it's such a pity that we have Brexit. It's a big pity, but it poses some advantages maybe to you with respect to the implementation of methods. And that's my question to, well, yeah, basically UKDNA working group, or Natural England, or whoever's involved in charge of implementing new methods. So now you don't have to comply with European Water Framework Directive, etc. Do you see this as an advantage, and are you moving the way towards implementation further?
Shall I say something, Si?
I think you're the best person to start with that one, Andy.
Okay. Well, firstly, I'm not really the best person, because Natural England doesn't do Water Framework Directive. That's a different agency. That's the Environment Agency. So our context is different. Yeah, it's interesting to reflect on this. I'm not going to try and justify Brexit in this context, but it does allow the UK to do some different things. And if I'm honest, one of the benefits of leaving the European Union is that we're no longer constrained by the Common Agricultural Policy. So we can really make some radical changes in how we are funding agricultural support systems and what we're paying for. So there's a risk in this.
Well, I think for the first time in the whole webinar, Andy's frozen a little bit, but we'll just we'll give him a couple of seconds to see if he comes back. And in the meantime, if Florian can just say boo, we can make sure that the webinar is working.
Boo.
Excellent. Okay. Well, Andy may bring us back. Obviously, I cannot see Willie here either, but Willie gave a presentation about that. Well, small pot of money to do some fact finding, and I think they're partnered with Nature Metrics as well. And well, hopefully some of the agencies are now thinking about what can be achieved in the context of DNA monitoring. And I think that was one of the drivers of the project that Willie was describing. But obviously, without extensive stakeholder representation, I don't know if Phil or Debbie or maybe Katie have any perspectives on that, given that Andy's Wi-Fi might have dropped out, or maybe the dog has chewed the cable.
I don't mind trying to say something, Si. It might not be quite what Andy was going to say, but I think it is something that through the DEFRA DNA Centre of Excellence we are really thinking about. And I think to what extent at the moment, you know, we can use these methods we haven't explored fully. I think there's a lot more work to do to identify those areas where we can use these types of methods in different ways, perhaps since leaving the EU. But at the moment, you know, we're just trying to focus on the opportunities that we do know we have and trying to sort of move those forward and get those methods really in operational use within all of the different agencies that are involved, I think.
Mm.
Yes, I'd go along with that. We're at the stage now, we've seen in a lot of the talks this week actually that we've got to move towards the routine monitoring in many cases.
Okay, and I know that I assume that the rationale behind the think piece is that I think Katie mentioned on the first day, that was kind of the DEFRA DNA Centre of Excellence wanting to reach out to a number of people to get their perspective on what could be achieved in relation to biodiversity assessment and ecosystem functioning, not necessarily following traditional technologies. And so there was a heavy focus there on how DNA and molecular techniques could deliver on that. But irrespective, obviously, Florian, as you alluded to, anything that does happen within the UK will always bounce off our EU colleagues because, well, we're obviously more powerful together. So whatever's going on will always be, be reaching out to the necessary actors in the field. So, okay, Kitty, are there any questions coming in?
There's been a few questions. The first one is, how should eDNA research be funded?
And that's a general question.
Mm-hmm.
That's a question that we've been trying to answer ever since 2014. But I would be interested to hear if anyone does have any additional additional insights into that.
I can say something, but I don't know where the hand is.
Oh, don't worry, Naira. We're going to use natural hands, if that's okay.
Okay.
I can't concentrate on you all and a little box out here. It'll be too much for me.
Okay. Well, maybe I can share my experience on fisheries management, not on eDNA, on population genetics, but our experience is that usually methods and results are easier uptaken when the research is actually funded by the agency or the body that needs this information. So this is not an answer to the question who should fund it, but it's more like if it's really funded by somebody that needs the result, it's going to be easier uptaken. So for example, in fisheries management, we have that, right? So when we have ICAT, International Commission for Conservation of Atlantic Tunas, or European DG Mare, or a body that really requires the information, they require us to do the analysis, for example, to assess connectivity within a fish species, then it's easier that this result is uptaken. If we do our research funded by, I don't know, our national agencies, just the normal competitive call, research call, and then we do the analysis, and then we knock at the door and we go like, okay, we have this result, maybe you can use it. It's more difficult that this result, it's really uptaken, or this method. This is just, if it helps.
I think that definitely does help, and I think something that we've learned along this journey is that if you want to do anything which is going to be of interest to end users, then the end users need to be present at the grant application stage. It's not a question of just knocking on the door even after you've got the grant. It's a question of co-design of the project. I think that is super important. And so trying to address those sort of co-funding streams would be really valuable in the future. And Kitty, just before we go back to any questions, Andy's come back.
I've come back. Yeah, I'm sorry about that at a power cut. I'm not going to come back to Florence's point, but so I'm not an academic and I'm not a researcher. So we've just sat through an amazing week of presentations. Is funding for research an issue? There seems to be a lot of it happening.
Well, that's a good question. Florian.
My impression is it's not an issue at the moment, depending on where you target. But if you look into the European calls, Horizon Europe, the new European funding, I find it almost crazy how often the term environmental DNA is even mentioned in the call. So often it's actually a requirement of the calls that you use and further develop the method. So at the moment, I see the method is widely accepted, and you can apply for money with more federal applied ministries here. They are happy to invest in further developing the methods, as well as National Science Foundation, as well as European funding. I have the impression it's absolutely no real problem. I think other research fields have much larger problems.
I think also reflecting on that, I suppose it all depends on what you want to get out at the end of the exercise. So I think just something that following on from what Katie was discussing. In the UK, I'm aware of a number of projects that have engaged with investigating DNA technologies, but potentially trying to get them to do the job that traditional technologies did. And hopefully we all know that that will not work, because it's asking a different sorts of screwdriver to open different sorts of, to undo different sorts of screws. So I do think that the end users and the agencies still have that really, really important role, that if they require a particular sort of tool or analysis, then that is also co-designed with the different actors in the research. I think that is really really important. And I think it probably depends on the funding environment you're looking at. I mean, from a UK perspective, UKRI Blue Skies funding streams, I think, are pretty tough to get this sort of work funded. But there will obviously be different funding streams out there. It all depends on whether or not the questions and the hypotheses being addressed in the research are directly aligned with the expectations of the stakeholders. I think that's quite an important thing. Okay, Kitty.
Before we move on, is it okay if we could stop sharing your screen size so everyone's face is like bigger?
Of course, yes. What can you see?
I can see your screen and your PowerPoint slide.
Oh, you're joking. I thought I could see you. I thought we could see you. You see you. Hang on. Here we go.
And there's been a few questions for Christine, so I've assigned them to you so you could go into the control panel and answer them. The next question is, how do we mobilize more of the Global South uptake into using eDNA methods, given these tend to be biodiversity hotspots, while also the most vulnerable to biodiversity loss?
Kitty, sorry, can you now see my screen? Can you see you lot?
No.
Oh, hang on. There. Can you see you all now?
No, I can still see your screen.
You've gone back to sharing your screen again. It was fine a minute ago.
I'll try and sort that out. And Kitty, because I was doing that, I missed the question. Sorry.
Yeah,
now you're fine.
repeat it. It says, how do we mobilize more of the Global South uptake into using eDNA methods, given these tend to be biodiversity hotspots, while also the most vulnerable to biodiversity loss?
So, Christy.
Yeah, so I think this is a really important question for our field because it's you know, so a lot of times when you drive something with technology, there's a kind of a double-edged sword. One is access to the technology itself. So in a lot of places, you know, like Christine was talking about, we need sort of kind of high-end laboratories and things like that for the research part, but we don't necessarily need that for when we want to roll it out, right? And so it's about creating access. And, you know, I think we heard some of the innovation coming out of the industry side, so these kind of container ship laboratories that we can start putting in places. And there's lots of ideas about getting out of the lab entirely. So a lot of the contamination stuff that we think about is actually because we take samples from a bunch of places and bring them into the lab. But if you're processing it in the field at that site, you know, what's the chance that you're going to contaminate it with another site? So, you know, it's like it's flipping it around a little bit. The other piece I think we have to do was a little bit the headlines that I showed in my talk is, you know, we have to separate the need of the people who need to understand their biodiversity from those who pay for it. So these people should not be the ones paying, you know, a company to make the data for them. They should be paid to take those samples, and that's how we have to flip it. We need to create jobs around this. We need to facilitate that they can be part of generating the data themselves without the burden that they're the ones bearing the cost of generating it, because they're already bearing the cost of the loss of the biodiversity. So we need to channel that investment in the global market that's coming into this and really find a financial mechanism to make that possible. So that's my ideas. There's some ideas out there, but we gotta, maybe we're not the right set of scientists. We need economists, we need other people playing a big role in our community to really figure out how to separate those two things from each other, the people who need the data from the people who pay for it.
Okay, Kitty. I'm still gonna try and share this screen a little bit better. But could we have another question, please?
Yeah, sure. The EA are still very much aligned to WFD, but agree that thinking is there about how and what we can do differently. Okay.
So do we have anyone comfortable with answering that one?
Can you repeat it once?
Yeah, I don't know if this is a comment or question, but they said the EA are still very aligned to WFD. But is there much we can do differently, or how we can do things differently?
I think that was Kerry from the Environment Agency put that in response to the earlier question that Florian was talking about, just to say that they are still aligned to WFD, but they are starting to think about how they might do things differently.
Okay. The next question is, population genetics of fish stocks have been used for a number of years, yet the practical use of DNA in management is still questioned. How can environmental DNA approaches overcome systematic issues like primer bias for conservation and management?
Christy or Mayara?
Yeah, I think one of the things that happens with eDNA is that we always tend to benchmark it against traditional data, for example. So, for example, when we use trawling data for fisheries management, trawling is also biased. It's not a primer bias, but it's a net bias, it's an area bias. So what happens is that we are obtaining different amounts of different types of information. So how should we deal with primer bias? The same way we deal with other types of bias in other types of samples that are not eDNA samples. Consider them. Of course, we cannot just ignore them, but that doesn't mean that eDNA data cannot be used because it has a bias, because the other data we're going to collect have also bias. Yeah, that's the message. It's like for my estuary analysis, the trawling data that has been collected for years and years and years is biased. The thing is that we assume that the bias is consistent, so we can compare. And now we change the method, the biases are going to be different. So we cannot continue the time series. We should start a new one, perhaps do at the same time trawling and eDNA. But the biases are going to be different, but both methods have bias. That's what I think.
And how are we doing for questions, Kitty?
There's quite about 5 or 6 more. Some of them. Does one of them asks, do you currently share your protocols online? And if so, where can I find them?
Is that a question to anyone in particular or everyone?
I think everyone. No.
I mean, I'm hoping that all of the published material, people have got transparent protocols, and I know that obviously the DNA Aquanet group have been working a lot on developing protocols with shared characteristics as well. I don't know if Florian wants to add to that.
Yeah, I guess so. We share all our protocols in the publications. DNA Aquanet did not produce kind of a cooking book with protocols, so the practical guide is more kind of like minimal standards decision making, but not like a cooking book. If you want to have this species, then you have to use that Taq polymerase. But I would say when I look into, well, let's say 70% of the research paper, probably I would say have sufficient details on the protocols in the supplement, etc. Yeah. And if they don't have, write to the authors.
I think that's a good point. Kitty?
This is another general question. We have seen this great progression in the application of eDNA approaches and companies that have been so pivotal in driving eDNA research. Judging from the talks, data and information sharing is high priority for all players, also the companies, which is great. But do you see some risk associated with the commercialization of eDNA research?
Maybe that's a question to any of the industry representatives. So, Alicia.
I'm talking from SpyGen point of view. We are kind of the first laboratory that sell eDNA analysis, and actually we and the other group like Applied Genomics and Naturmetrics actually contribute to the scientific spread of environmental DNA. So from our point of view, we use our income to finance the research that we produce. So there is a trade-off, and as we are not academics, we need money to do our research. So it doesn't really matter the selling the eDNA. We are completely transparent and we are fair with our client as the other company do on eDNA. So I don't see the problem.
Sai, if I could jump in there quick, I would be happy to. Yeah, I mean, somebody who's, straddling this right now, I mean, I'm a professor and I just started a company. And, for me, it's all about creating access. There's only so much I can do in my academic lab. And, if I didn't press upon everybody to think about the gravity of what we're facing with our planet, like if we can get anybody out there doing this anywhere, we got to mobilize all sectors of humanity to do this. So there's no conflict of interest of paying for a service if somebody gets data, gets what they want for that service. So, you know, and there's lots of ways to get that. And, you know, what's fascinating and humbling about being in a company setting is, you know, it can get done so much faster. Really, the time scale. If you go in an academic lab, you know, I mean, it's wonderful. We train students. We can do novel things. We can advance the technology, but it's slow because we're learning, you know, and the stuff that's ready to be out and transmitted and used right now, it needs to be upscaled, and we need government labs, we need private sector, we need it all out there because there's no time to waste. So yeah, I mean, you know, and it's great that there's all, you know, there's, you know, a few companies here represented, but I posted a question on ResearchGate some years ago, and there's over 50 companies all over the world now offering these as services, you know, and there is this regional thing. It's creating jobs, it's giving our students jobs, it's, you know, so it's incredible for the economy. And yeah, so I don't know, I don't see a conflict. There's a lot of questions with, you know, is DNA a commercial good? You know, there's some legal things that we really need to grapple with. How do we deal with Nagoya Protocol? How do we, you know, create sovereignty over data? Those, I think, are more important questions than who's generating it.
Thanks for that, Christy.
There's a few more questions, but they're quite specific to like protocols. The first one is, which is the best way to preserve eDNA?
Cold, dry, and buffers. I think those are the three go-to methods. I guess
Section
it all depends.
2.3 of the practical guide will tell you exactly.
Excellent, Christy. So yeah, hopefully that'll be good. If obviously people will be able to address any sort of super technical questions. I've got a question, if you don't mind, Kitty. And so this is a general question, and I'm kind of reflecting on the DNA working group journey and where we are with the different achievements, and also reflecting back on speed, and also reflecting back on impatience and biodiversity loss and how we can do things better. I find it absolutely fascinating that the industry representatives are generating a great deal of business with different customers that require biodiversity data to be generated, that presumably, you know, they will pay for. And there seems to be, you know, a great deal of work going along those lines. But yet on the sort of like regulatory and the statutory side, and this is alluding to Florian's talk as well, I guess, you know, what many of us are here to do is to try and enact that change, so that we can work with regulators and those sorts of end users and stakeholders. And so we've had these discussions over many years, and so I guess really from any of the sort of stakeholder representatives, in an ideal world, what do you think we need to do now in order to start accelerating the uptake of DNA technologies? I mean, we're doing well, and we're getting all sorts of different structures in place and making good progress. But what more should we be doing as a grouping?
Well, is that me? Well, it's Mayor Katie or Debbie, I think, are here as
you can take it in turn.
Government end users. So a couple of reflections on that. I mean, the first is that I think it is happening. You know, I haven't been involved in this group for as long as you, but I have been involved for a fair few years now. If I think back in the first meeting that I went to and where we are now, it has changed an awful lot. The whole sector has matured. The view within governments and within government agencies has matured and changed. So I think it's gone from within our agencies, gone from a view of what on earth is that? Is it magic? To an acceptance of these techniques and an eagerness to use them, but an unfamiliar, still some caution. So I think that's the first one. I think it's, just changed immensely, and that change is happening within this sector. The second one is to go back to Ryan's presentation, which is to make it boring, or from our point of view, it is now into the realms of taking individual applications for, whether it's for assemblages or for individual species assays, and just going through that boring process of verification, validation, limits of detection. So we've got all that set down, and people can just, our staff with a fairly basic knowledge can take this off the shelf, commission contracts from consultancies, and be confident in using the results. So I'm afraid it's that boring bit that we have to do now.
And so how does that, I'm looking at Florian, even though I may not be sharing my screen that effectively, and you may not be able to see I'm looking at him. How does that talk to your ring testing exercise, Florian, that you did? Something along those lines.
Well, actually, lots of ring tests happened and are happening. And so both internationally, on the one hand, via DNAquanet, and the diatom results are super encouraging, and 19 labs, or how many participated, and I think it looks all very good. The macrozobenthos analysis is ongoing, no results there yet. Another European project presented results that it's very robust. So there's variation at taxa lists, but not at index, or index level results are very robust. So my impression is, I have no clue actually how many exercises were done in the past for the traditional methods until a method was accepted. But my impression is the robustness of the present methods has now been shown quite a bunch of times. So we're doing also national tests. So I actually have no experience how often you have to show something or prove something or document the variation. It's never perfect. It's never 100%. So it's probably like a 10% variation. But this variation has been quantified for the traditional indices as well, right? So I have no experience how it was in the past until something gets accepted. So I cannot say how much more is needed. I would definitely say there's room for much more tests, yes, for sure. But all that I see is already, it's never the same, but it's quite robust, what we see.
I think it depends on the application as well. Now, I go back, so another reflection in this week is it might be the first one that we haven't talked about great crested newts. And so that's exactly. So, but that perhaps is a demonstration of the fact that that's just got to that boring stage where we're just using it routinely.
It's a good point. It's a good point. I didn't actually have that in my notes, Randy. I think that's very insightful. I don't think I saw a single newt in these presentations. Okay, Kitty, we've got 2 minutes. So have you got any emerging questions that you'd like to ask the audience or the panel?
There's still some very protocol-specific questions. I don't know if I should ask that. Oh, there's another question that asks, what are the main criteria for an eDNA paper to be accepted when you're reviewing submissions?
Wow, that's one for Christy, I think.
Yeah, at the journal, we don't have a filter of novelty. So I really want to emphasize that. And that's what I meant in my talk. We need demonstration after demonstration so that we make this boring. and so there isn't that criteria. What we look for is robustness, and a lot of what Kristen talked about earlier, you know, that we don't have an amazing insight because it's a false positive or a false, you know, something done there. So we really take a really good look at, you know, where negative controls used and things like that. And we do reject papers that really don't follow basic sort of validation criteria. That is the floor for us. And beyond that, we're, you know, the floodgate is open, as you noticed. So, you know, just making sure that those are robust. And, you know, we usually give the benefit of the doubt. If it's not in there, it goes out for review, and, you know, people can comment and revise, because, you know, just because it's not in the manuscript at the first time someone sees it doesn't mean you didn't do it. And so it's often, it just got lost in all the details, and it needs to be highlighted, the robustness of the methods. And yeah, we have conversations at the editorial level, but, you know, some of it is, it's just basic things that we've set out for ourselves as a field, and yeah, so, but not a novelty thing, in my opinion.
Okay, thank you, Christy. And just echoing that, obviously, when anyone's designing experiments, just think about those controls. Christine mentions that using controls that you don't expect to find in your study, and they can be immensely valuable when it comes to filtering your data. We're past 2:30 now, GMT time, and so I will just extend my thanks again to the organising committee, all of the speakers, and everyone who's joined the panel today. This has been a bit of a blur for me this week, but the fact that some very mature and very well-respected colleagues have all used the same phrases as amazing to reflect on what they've seen this week, I think that's a testament to everyone's hard work. So I think I'm still in charge of the webinar, so I'll sign off and thank you everyone for your contributions, and hopefully next time it'll be in a bar with a drink, in person, post-COVID, and you know, it will be good. Okay, I'm going to sign off.
Thank you.
Thank you, everyone.
Thank
Thank
you.
you.
Thank you, Sai.
Thanks, Sai.
Bye bye.